I can hardly wait to get back from lunch to hear all about bioinformatics data pipelines. And I'm so glad I've been able to give you what you were looking for on the way to lunch. So what I'm going to do, spend the next 20, 25 minutes talking about, is some of our experiences directly within the Blueprint project, and also within the Ensemble project, where which is also participating in Blueprint, for how we're collecting, managing, doing uniform processing, and then integrative analysis on specifically on the Blueprint data and data types like it, for example, like Encode. Obviously, this represents just a small piece of the informatics working of the Blueprint project, and indeed a very small piece of the type of iHack or other efforts. But I hope what I talk about is a useful example and has useful information about how this works at the scale. So I'm going to start talking about Blueprint data coordination. To remind everyone, Blueprint is collecting data on blood cells, on the epigenome of blood, and how this and how it differentiates. So once data is generated, and fortunately I don't have to discuss any of those details, once data is generated from a sample somewhere in this cascade, we bring it into the EBI from the data generation group. And basically we have a situation where there are 3 sequencing groups, and these groups produce data. That data is then submitted to the raw data, submitted to our protective database at the EBI, which is called the European Genome Phenome Archive. For those of you that haven't interacted with it before, it's most similar to the dbGaP database at the NCBI, and it provides a layer of data security for data that is protected by consent agreements from the individuals who provided the samples. So that there's a process for data submission, then that ends up inside the EGA data archive. For those of you that are wondering, the EGA data archive is actually physically separated from the other parts of the EBI. We have a section of this archive that we call the vault. The vault is the secure area where the data is stored. It's disconnected from the internet and disconnected from other parts of the EBI. So it's essentially protected. In fact, it's completely protected. We take data security very seriously here. Although it's the mission of the EBI to provide data out to the widest possible grouping, the goal of the EGA is to make sure that the data only goes out to those people who are supposed to get it. So data coming out of the EGA in general goes through a data access committee. The Blueprint project has a data access committee made up of some of the work package leaders and other people from the project. And that data access committee would receive applications from people who want to use the data, and then once those applications are approved, the data is provided from the EGA to the approved researchers. Of course, data is also provided to the consortium members as part of the analysis that we want to do within the project. So one of the things that we've been able to do in the context of the Blueprint project is take advantage of the existing resources that we have at the EBI, where there's considerable investment in and considerable expertise base. So the EGA uses a lot of the infrastructure of the European Nucleotide Archive, and we also use concepts in the Array Express archive. So we've really been able to leverage a lot of the things that exist at the EBI to provide this service to the Blueprint project. So bringing the data in, we rely across the project on a distributed method. The sequencing centers conduct their own QC, so the data that they send us passes their own internal filters. This, of course, makes sense. The sequencing centers need to know what they have produced and what their data actually looks like. But to ensure a uniformity, we also, once we get the data in-house, we run a series of quality control and basic checks on this to make sure that we understand the data that's come to us as well. Now, this doesn't in any way substitute or try to override what the sequencing centers do. They are, of course, experts in the data that they've produced. But this allows us to understand at a basic level to make sure nothing unusual has happened, and we're providing a baseline that we can provide to other members of the consortium. We also run some so-called pre-alignment QC, which is developed at a group from the Babraham Institute, which is not too far away from us in Cambridge. And this, again, gives us some basic understanding about the raw data that we get in. So once we've gone through that basic level of quality control, we have some standard pipelines that we run data through. I'll go through some of those on the next couple of slides. And we have different types of processes that take place within Blueprint that are specific to different data types. And these are, in fact, run at different places around the project in such a way that, again, we develop expertise within groups on the project. So the ChIP-seq and the DNA-seq data gets done at the EBI, while the RNA-seq data gets done in Barcelona at the CRG, and the whole genome bisulfite sequencing data is done at the Spanish National Sequencing Center, which is also in Barcelona. So basically to provide a little more detail here, I'm not going to go deeply into detail. Unfortunately, I know many of you were wanting to see the detailed command lines of the bioinformatics tools that we use and all the various parameters. I'm not going to provide that, so I'm sorry to disappoint the people that wanted that. If you are interested, please come and see me later, or come and see Laura or Dave, and we can provide all of those. So to start with, the ChIP-seq and DNA-seq, we use the same pipeline for both of these data types to the extent that we can, which is that the raw sequencing data is basically split up into smaller chunks so we can put it across the compute farm. And then we run the BWA alignment program to basically align them all to the genomes that we're using. We take the final alignments, which again come off the different machines of the compute farm, we merge them all together, and we produce a single alignment file. That's this BAM file, which is a binary alignment file. This process basically allows us to do things very quickly, but also if one of these jobs happens to fail, it's easy to pick up the part that has failed, rerun that, and put everything together in a very, very quick way. So it's a very robust approach. So once we've finished the alignment, again, here's the BAM file that ended the last slide. We bring it down through a number of post-processing steps. So basically looking at where we see duplicates in the file, some other metrics, again, QC, and then we have a final file that contains all of the data that's gone through this process. I think it's also important to note that we're not removing any data here. We're just marking it appropriately so people know how it's been aligned and what is there. So all of the data is still available. It could be reanalyzed by another group, but we've given it our pass analysis. So again, once we go through once we have this BAM file, we take this now we start to break it apart depending on the different types of analysis that we're doing. And we have the ChIP-seq analysis. ChIP-seq goes through a program called MAX. MAX is a common peak calling program that we have a lot of experience with, lots of other people have a lot of experience with. We have very good reason to believe that it is robust across a wide variety of situations. If you're interested, I could share some of our internal quality testing and other things that we've done to make us believe in a program like MAX. But again, compared to other programs that we've gone out and tested, we like the characteristics of MAX. DNA-seq, of course, is a little bit different. It has other characteristics, both in the way the data is distributed across the genome, but also the length of the sequencing reads and a few other things, the expected size of, for example, the regions that you find and things like that. So we use a program called Hotspot to do that analysis. And from those two, then we get a set of regions, the ones that are positive for DNA-seq, the ones that are positive for ChIP-seq. This goes into a bed file, which is just a file format that marks regions across the genome. And then we can compute information about those regions, how close they are to transcription start sites, how they overlap other features in the genome, things of that sort. But again, as I mentioned before, all of the data exists back here, and so if there are other people who want to reanalyze it in a different way, and indeed, if it turns out in the future that there are different programs that are more effective at analyzing the data, either at this step or farther up in the alignment step, we could conceivably recreate these files. We could conceivably rerun them and produce updated files. One of the things that I think is important here, and important generally, is that we recognize that the process of analyzing epigenetic data is not in any way finalized. This is not a static process. This is not something that is as well understood as measuring someone's weight, which is actually, if you think about it, that's pretty stable technology. You can believe in the weight measurements that were 100 years ago, or maybe 200 years ago, or maybe longer. I don't think you can believe in the epigenetic measurements that we took 100 years ago. And you won't be able to use our current methodology for the epigenetic experiments we take 100 years from now. So data distribution then. So I should say, you will remember I noted that there were different types of analysis that were done at Barcelona, at basically the 2 institutes in Barcelona. I'm not going to go through those in detail, only because there is some chance that I would have many of the details wrong. And if you're interested, at least for part of those, I think Simon is here in the audience for the DNA methylation data. I'm not sure who is currently here representing the RNA-seq data at CRG, but these would have gone through a similar process that looks very like this in that there are checkpoints and quality control. They use different programs and different opinions. So when we get to data distribution, we have in Blueprint 2 main classes of data. What we call our individually unique data. This is reads and alignments. So this is data that has, for example, the SNPs within it, because they're from the DNA sequencing reads, of the original donors. In theory, this data, combined with another measure of that person's genome sequence, be it genotyping arrays or something else, could be used to match the data that was collected in Blueprint to this other data that is from the same individual, and we could link that data together. So we have the individually unique data and the so-called non-unique data. These are things like enriched regions, so the regions that are positive for DNase hypersensitivity, quantifications, metrics, other things. Now, some of you know, of course, that the boundary between unique and non-unique data is not a permanent one, and in fact, it has people's assumptions of what was unique and non-unique data have changed over time. That could happen again, but this is our current understanding. All of the individually unique data is distributed through the EGA. Again, this is the secure archive where we store the data securely and we provide it out only with people applying through data access agreements. The non-unique data is distributed in a number of ways. As soon as we can, we provide some of this on a private FTP site for project use. And we also have phased public releases. Our goal is to get this data out for the public to use absolutely as soon as possible. But we do need to have some internal quality checks within the project to make sure that what we're releasing is more useful than not. And that's what the so-called private site is, so that we can share things amongst ourselves until we're sure that it's right. So what data is available? I think this is still correct. We have 11 donors in total, roughly split between male and female, and a host of different experimental types that I've described already. DNase hypersensitivity, ChIP-seq, RNA-seq, and so on. So the non-unique data files, we provide these really right on this FTP site. We also have a host of file formats, and critically we provide them as a track hub. Track hubs provide an ability for data to be visualized on both the UCSC and the Ensemble genome browsers. There's been other discussion at this meeting already about how one can put additional information on top of track hubs to access data in both visualization ways and programmatic ways. We've found the track hub concept to be very useful, and we think it will be very useful in the community as we extend it further, including in some specific ways that I think will be useful for IHEC, ways that may incorporate things like the IHEC metadata or other aspects that we haven't completely explored yet. So to give you an example of what this looks like, here's a whole host of data coming out of the Track Hub. This is all on the Ensemble genome browser. I think it's probably true. Most of you will have seen Track Hub data in the past displayed on the UCSC genome browser. But given that, I don't think it's a big stretch that you can see that this is basically the same type of display, where we have genes down here and the various types of histone modifications and other information spread across in various tracks. And many things, although it's a little bit challenging to see it this zoomed out, but you can see that many things will end up lining up around important features of genes. Transcription start sites, the gene's bodies, things of that sort. But we don't just provide this data visualization and raw data file download. We also provide methods to do specific data mining across the Blueprint data sets. This is based on the Biomart technology. For those of you who are not familiar, Biomart is a method to do structured queries across databases. So what does structured queries across databases mean? It means you can ask questions like, I want all cell types of a certain type, or I want all data from a certain cell type that displays a certain mark. And you can ask that question, and the software will bring it back to you. This is the, admittedly, not very exciting user interface that one uses to ask those questions, where we have certain types. You may not be able to see this, but there's certain types of analysis, people who've done the analysis, sample IDs, and so on. And this information can then be selected appropriately. You define what you would like the output to be, containing information that we have in the database. So here, for example, we've selected the gene stable ID, where it's located in the genome, as well as the normalized expression level. And this information will bring you back information about the RNA-seq experiment, indeed about the analyzed RNA-seq experiment that we've stored in this Blueprint, that we've generated in Blueprint and stored in this Biomart data mining interface. This is the type of information that then comes out. Now, for some people, this is, of course, not very useful because it's just lines and lines of text. But for people like bioinformaticians who want to do the analysis on this, this is a near ideal format, where you come out with data organized by analysis ID and the information that you want to do computation on. So we also, in the Blueprint project, provide data also for browsing via Genomatics and the Genomatics browser. Genomatics is one of the SMEs in the Blueprint project, and you can see they have a dedicated website. Genomatics is also, in fact, reasonably well known across Europe and beyond as one of the very important bioinformatics service companies that provide unique ways to look at and analyze data. And you can see here, this is, again, regions where we have different data types with the genes on this track. And this is another way to approach and get in and understand, visualize, and use the data that we've generated in the Blueprint project. So over the last few minutes, I'm going to talk about Ensemble integrative analysis. So up to this point, I've talked about basically the raw data that we've taken in the Blueprint project. We've done analysis to find, say, regions of the genome that are enriched for something or other, be it ChIP-seq or DNA-seq. Within the Ensemble integrative analysis, we are trying to turn all this information into understanding of how the genome is functioning at these regions. This process, we've been working on it within Ensemble now for actually several years. It's called the Ensemble regulatory build. And the goal here is to basically annotate the whole genome and tell you what the genome is doing in given cell types. And in fact, more generally, tell you what the genome is doing overall. This process has developed over time. We've incorporated new informatics innovations like whole genome segmentation that I'll talk about in just a minute. And the goal, again, is to assign this fixed function to genomic regions across all cell types and do that in both a cell type and a multicellular cellular way. And finally, to come up with something that, again, is useful, is rigorous and robust, but is useful for downstream analyses. When people want to, for example, analyze variants that they've discovered in a patient with a given disease, or they want to analyze GWAS hits, they're not interested in comparing this individually to 27 different marks across the genome or the epigenome. They're interested in comparing this to an annotation that says, that's a predicted enhancer. And we want to be able to facilitate that analysis. So I talked a little bit about the Encode segmentation algorithms. These are computational methods that were developed recently and part of the Encode project, which was published in 2012. Of course, these algorithms go back a little bit further than that. There were two main algorithms used, Chrome-HMM, which was from Manolis Kellis's group, and Segway, which was from Bill Noble's group at the University of Washington. They have a number of different characteristics to get to the same thing, which is, again, a division of the genome into different states by incorporating multiple different epigenetic types. For those of you that know the difference between a hidden Markov model and a dynamic Bayesian network, you understand why you might want to use one or the other. For those of you who don't understand the difference, it doesn't really matter maybe. There's different resolutions on the genome. There's different details, all of this. Critical things, though, when you take a step back and think about how one might want to deal with this, to understand what happens when data is missing, for example, or how one handles the length of given states, we understand that the genome has different lengths when it has different features. These are all modeled in specific ways. And it's important to understand how this is done when one wants to do a full integrative analysis. So I'll leave that summary here. Obviously, for those of you who want more information about these, both of these are published methods and are relatively widely used. This is the type of information that comes out of this, where we have different signals. So here we have different histone modifications. Here we have CTCF, DNase hypersensitivity. This is, of course, all from Encode data. And here's the different types of states that come out of this. And you can see, for example, things that you already know. Things like transcription start sites have histone H3K4 trimethylation, for example, which is a well-known marker of transcription start sites. And there's a whole host of other pieces of information that show up inside these state diagrams that when combined together, and even when you view individually, you can learn a lot about what the genome's doing. So we use this information then across the whole genome and produce our final understanding of what the genome is trying to tell us, at least from these data. So if you look at this, here's a whole host of different cell types. Up at the top here, we've done Chrome-HMM, which is one of those two algorithms. Down here, we've done the Segway, which is the other algorithm. And you can see the cell lines repeat. Here's the GM12878, the lymphoblastoid cell line. Here it is again. It's done with Chrome-HMM. It's done with Segway. And you'll notice immediately, actually, that there's real differences in these two segmentation algorithms. The Chrome-HMM is more blocky. The Segway is more, it's more noisy, but it's also more detailed. And so we set about trying to find a way that we can integrate this and come up with a single measure of what we're looking at. Here we've done it for CTCF, for insulators. And what we're doing is basically summing this information from the above tracks into something that looks very like a wiggle track down here at the bottom, but in fact represents all of these cell types viewed together. And so this, for example, is a CTCF site that appears in only a few cell types, where these CTCF sites appear in all of them. They're much stronger. They're much more common across various cell types. We can do the same type of thing with transcription start sites. These are cell type specific transcription start sites. Here's ubiquitous transcription start sites. And from this, present an integrated approach to how the genome function works. Enhancers are potentially more interesting because enhancers are much more likely to be cell or tissue type specific. And you can see, again, we have more complex patterns when we get all of this together in this way. Promoters, too, have interesting characteristics where we've, again, followed this same approach. And now if we put the genes track down here to make this more clear, we can see how clearly the promoters from the regulatory build line up against the known genes that we have in this region. Lastly, we can also do this with transcription. And you can see here this region is highly transcribed. Here's another region that's highly transcribed. Both of these sit over genes. Here's one of the genes. Here's one of the other genes. But of course, although one can predict transcription from the epigenome state, one predicts transcription a lot better from RNA sequencing. And so rather than try to tie ourselves into knots and try to predict transcription from the epigenetic state, we should just use the RNA-seq as a prediction of transcription, because in fact that's a direct measurement and that's what we want to get. So when we put it all together, we then have this now view of the genome where we have transcription start sites, promoters, insulators, enhancers, and of course we also have this, this measure of transcription, which we believe is actually replaceable by the RNA-seq data. So in giving this, we have then a nice view of what the genome is doing. This is very likely to be correct, and I know I'm hitting my limit here. We've looked at a number of different ways that we can convince ourselves this is correct by looking at VISTA enhancers, P300 binding assays from Encode, as well as data that was generated from Phantom 4, where we have cage tags and tag clusters. Here's what the cage tag analysis looks like. But all of this brings us to the conclusion that, in fact, this type of integrative analysis is both conceptually straightforward, easy to compute, and has the advantages of being both good recall and reproducible across multiple cell lines from the places where we can do the validation. And so with that, I do want to acknowledge the people that actually do this. Many of these people are sitting in the room, including Laura, Dave, Steve Wilder, and Daniel Zabino. But of course, there's other people at the EBI that make this possible. So with that, thank you very much.
How well do the marks for transcription you predict from the segmentation correlate with the actual RNA-seq data?
So essentially the correlation is very, very good. The RNA-seq data has additional information rather than just this whole region. The RNA-seq data has information about splicing and other things that are lost when one looks at the histone modifications. I don't know, do we have a quantification of how many times we have, I'm kind of looking at Daniel and he's kind of looking away. I don't think we've actually done that quantification, but when you look at it by eye, it's very clear that it matches up. Yeah. So clearly that's the type of information that would be useful for me to have on that slide and that I don't have to hand.